Let's go on to Drew Endy, Stanford University, building a biofab.
Thanks, Sam. I guess I'd agree that synthetic biology per se is not dangerous, but your slides remind me that people can be dangerous in groups. I spent some time this summer in Estonia, where folks in my family fled at the end of World War II to come to the United States ahead of the second Soviet occupation, and returning to Parita outside of Tallinn and seeing the students who were learning how to sail for the first time in generations because much of the sailing was disallowed under the Soviet occupation for the fact that the newly learned sailors would head off to Finland. Just absolutely remarkable how people can choose to do things which are incredibly destructive. And so a very inspiring set of remarks. I want to talk about building a biofab. This is something that I've mentioned before and continues to make incremental progress. My remarks will probably go for 15 minutes, and I'll end with 2 challenges or questions or topics for discussion. A way to come into this is to go to a talk from yesterday. This is the research paper which is coupled to a lot of the advances in Amyris biotechnologies. We heard how at Amyris they've been able to take forward the work of the Kiesling lab to produce artemisinic acid for treating malaria, replacing a supply of this chemical instead of harvesting it from the bark of a wormwood tree, growing it in a vat of yeast or what have you. This is a project that looks terrific and is a huge success and absolutely should be celebrated. But from an operational perspective, you could evaluate it in terms of its costs. Its costs include approximately $100 million US of research funding, both from the Gates Foundation and elsewhere, probably over 100, if not 200 postdoc years, experts spending a tremendous amount of time trying to figure out how to get this to work, and so on. And so one of the questions we face in synthetic biology, since we are 35 years, if not more, into synthetic biology, is how do we take a project like this, and instead of having it cost $100 million and of order a decade of work, how do we make it a $10 million project, a $1 million project, a $10,000 project, a project that an undergraduate might lay out on a computer workstation at some point in the future and have it actually go? So I think that's one of the challenges we as a field face if we want to come back to a future 5 years from now, 10 years from now, that's actually radically different, revolutionarily changed from where we are today. It would be absolutely dissatisfying to imagine coming back to synthetic biology 10.0 and seeing yet another talk on, the 236 amino acids of lambda repressor. God bless lambda repressor. There are experiences whereby human beings have gone through transitions that make things like this possible, and it's an open question as to whether or not we might be able to do the same for biotechnology. So, for example, I'm trained not as a chemical engineer, but as a structural engineer. And so in my intellectual history, I've been familiar with building things out of objects like what you see here. Now, these sorts of objects are natural objects. And these rocks are very useful in terms of things you can make stuff with. You could build a wall or a dam or what have you. But all these rocks we find in nature are very interesting. They're all so different. And if you wanted to, you could go off and study the rockiome and become a geologist, right? And that would be a nice, good thing to do. But in terms of building, a question comes, how do we make this substrate a little more engineerable? And so at some point, a different group, the builders, decided to invest some of their energy in tools that make more regular, standardized rocks. Later on, in the Stone Age, folks started putting these things together and making incredible artifacts like cathedrals. Now, if you think about it, in Europe, a lot of the cathedrals weren't built in 7 days. They weren't built in 7 weeks. They weren't built in 7 years. They were built in 70 years or hundreds of years, right? Not as bad as the Artemisinin project, but representative of what happens in a technology where you don't have tools that might help you with the work. Later on in the Stone Age, getting more towards where we are, obviously we've somehow figured out how to construct things, how to design things, how to actually put things up. Here's the Hong Kong skyline. This doesn't take 600 years to make in this form, right? So how do we get to something like this? Well, the question I've been thinking about is not so much what are the applications of synthetic biology, because I'm convinced that there's more than any of us could imagine, and we could occupy tens of meetings and never come up with the limitation exactly as was quoted in the earlier paper from 30 years ago. I think the focus needs to be in part on technology platforms that enable the design, technology platforms that enable construction, and technology platforms that facilitate characterization. So I'll come to each of these in turn. What's great about the field is that there's been a tremendous amount of progress in each of these. So for example, here's a project from 2004. This is before Synthetic Biology 1.0. Jonathan Goler in Tom Knight's lab was a master's student, and in less than a year, he put together this thing, BioJade, a design and simulation tool for synthetic biological systems. And if you went to this tool, you'd run the program. It gives you a layout screen on your computer. There's a little double helix here, which is a helper thing if you're familiar with Microsoft Office, and the paperclip Clippy. You know, this was Jonathan's attempt to provide a little double helix helper for you. You could lay out some abstract devices, stamp them, connect them up, send it off to a detailed molecular simulator that would track single molecule reaction events and simulate the dynamics of your system. Synchronize with the then nascent registry of standard biological parts, calling into that database, getting access to the DNA sequences, assembling an integrated genetic construct from that, using some layout scheme here, right? So this is a technology platform that's actually 4 years old, which exists, right? And so it's interesting to note that very few of us today, certainly not myself, are using an actual platform like this when we go to design any of our constructs, right? This hasn't actually translated from a notion, something that can be demonstrated and is obviously going to work on a computer, to something that goes into practice either in labs or in industry. Moving on to construction, building on Rob Carlson's plot here, I'll just load it all up and then we can talk through it. Rob noted that the ability to construct oligonucleotides, short fragments of DNA of order 200 bases long, has increased quite significantly from about 1990 through today and going forward. Now, the chemistry of Carruthers gets worked out around 1982, so far as I can figure, for phosphoramidite synthesis-based chemistry. If this is where we're going in terms of synthesis, how do we get still much better at it? The fact is today, if you went to order a gene, and let's say it was 10,000 base pairs long, you'd be rolling the dice in terms of whether or not that would come back within a month, and whether or not it would come back at all, and how it might have to be debugged. And I thought that the M-Genitalium Genome Construction Project had a nice data set in the paper that came out from Venture Institute on all the sorts of errors that they encountered in that construction project. But if you look at DNA construction, this is the construction technology we might want to get better at, you can separate it as a function of length scales. So the raw materials, right? The raw materials, for example, are currently derived from sugarcane, having previously been produced from salmon milk. So that sounds pretty good. Sugarcane is a commodity, and so long as the biofuels groups don't eat it all up for us, we'll still be able to synthesize DNA. The phosphoramidite reagents are okay. Oligos is interesting. The best oligo synthesizers I know about come from Agilent. It's an inkjet-based technology. They claim to be able to make 55,000 oligonucleotides of length 200 long with an error rate of 1 in 250. So that's a 10 million base synthesis run. That's almost the size of a yeast genome. It'd be interesting to imagine moving everything from the oligo length scale down to the bottom inside cells, right? It's not that these commodities are bad per se, but imagine if you didn't have to buy the chemicals. Imagine if you could do the de novo synthesis directly inside a cell. So if you could imagine each of these length scales as I come to them, I think what I'm getting at is there's an opportunity for us as a community to outline and execute along a roadmap that would make our construction technologies much more powerful. So again, for example, I think there should be research projects at the oligo level and below to move DNA synthesis into living organisms using the native pools of reagents to do the construction. Whether this might involve modifying a terminal transferase or something that allows for template-independent DNA synthesis would be an interesting thing to consider. Moving up above oligos to genes, things up to 10 kb, 100 kb, what have you, and then genomes above 500 kb, it's quite apparent that we're in a weird situation. On the one hand, we've got a commodity marketplace for customized products. Gene construction is basically a commodity business where the price since SB 1.0 has collapsed from about $4 a base pair to under $1 a base pair, But if you're running a factory that's making a custom product on a commodity basis, if the specific request from any one customer has a problem within your process, you've got a tremendous downstream cost associated with rework and figuring out how come you can't make that thing. So anybody who could, for example, get better at delivering a 10 kb fragment of DNA within 24 hours would be doing a world service. What's very interesting to note, speaking about the United States, is I know of no such focused effort to deliver that technology. The last I heard, there was a program in 2003 sponsored by DARPA to do just that, but it was canceled. It's crazy to me that if you think about what our promises are around synthetic biology, we don't have a better structuring of the investments, a public-private partnership, to go back to yesterday's afternoon session, around how to make some of these foundational tools we need, like construction, better. The genome construction projects are beautiful works of art today. It's not an accessible technology. Ataya's group at Mitsubishi, the Venture Institute group, those are great demonstrations, but they're not things that take matters of weeks yet, so far as I've seen. Going from Ataya and a serial process, 7 years to make a 7.7 million base pair fragment, to a logarithmic process from Dan Gibson et al. We're still looking at opportunities to do parallel assembly of genome-length fragments of DNA. And again, there's tremendous opportunities for improvements in our basic construction tools. In terms of characterization, one way to evaluate how we're doing with characterization is to look at this registry of standard biological parts, this pilot project that's spun out of MIT. So on the superficial level, it looks awesome. There are 2,000 BioBrick parts today. They're freely available. And if you think about that, 2,000 parts of average length 500 means that there's an aggregate set of DNA there of length 1 million base pairs. And if you had to go build that yourself, the cost to you would be about $1 million. The fact that via iGEM, teams of students are getting these books for costs that add up to about $1,000 is a huge feat. But if you go into this, what you'll see is there's some incredible things, like a banana odor generator, a light generator, a fluorescent protein generator. Here's a cell-cell communication receiver device. These are some of the best characterized components within the registry. And if you look just at this one, this is a project that Barry Canton and Anya Labnow at Ginkgo Bioworks and Berkeley Biophysics, respectively now, did at MIT. It took them of order 2 person years to produce the description of this cell-cell communication receiver device shown on this data sheet. Things like the input-output relationship, the latency, the input compatibility, what it will respond to in terms of different chemicals, the evolutionary stability of this construct, how quickly it will mutate in the presence of different input signals, the demand it places on the cellular chassis, how many charged tRNA per second it's expected to consume if you wanted to bring this into a model, as per what Christalla Jones-Prather was discussing yesterday. So that's pretty nice. And we've got a process for making many of these, if you will. But if you then come back and look at most of the parts in the registry, those that are produced by this extraordinary group of people, the iGEM students, you'll find things like the Melbourne, Australian team who made I-750016, this gas vesicle polycistronic gene, that when you put the DNA into the cell, will boot up a little protein balloon causing the cell to float or sink, depending on the number. But the actual operation of this isn't so well characterized. And if you look at many of the parts, in fact, most of the parts in the registry, you'll see something like this. So this is transcriptional terminators, things that stop RNA polymerase from trundling along DNA. One of the most basic building blocks you'd want to use to write any genetic program in bacteria. There are about, 30 of these. And this green A means that all these transcriptional terminators are available. They've got nice little numbers here. This next box describes the functional status of these terminators. So for the green W, that means somebody's used it or characterized it and is gone on record as saying it works. And then the red X is it doesn't work. And then the null, the empty box, means nobody's looked. Somebody's done the work to design the DNA and made it available, and nobody's ever tried it out, or if they have tried it out, they haven't come back and entered in the information. And then if you look at the ones that work and come across, in these next to last 2 columns, you can see what the goodness of the terminators are. This is the efficiency of termination. That is, for every molecule of RNA polymerase that comes along the DNA, how many get kicked off? right? And if you had a rocking terminator, it'd be 100%, right? Well, this first one is 42% in the forward direction, and then in the backwards direction, it's 64%. Okay. This second one, 60% in the forward direction, and nobody's ever looked in the backward direction. Use it at your own risk. This next one, B below 15, 98% in the forward direction. Pretty good. 30% in the backwards direction. Not so good. And so on. For folks who are familiar with the registry and have made constructs from this collection that are larger using some of these, it turns out that B below 15, just as an example, is one of our most popular terminators. Meaning you see it used all over the place. Oftentimes you'll see it used multiple times in the same construct. And what that leads to practically is often direct repeats along a higher order DNA construct, leading to looping and deletion over time. This is for transcriptional terminators, some of the simplest things one could hope to use. As a technology base, as a set of tools, we're not looking so good, to be honest. And so this leads to the idea that we might create a new community resource, which could be called the BioFab. And the BioFab is meant to be a production facility for making high-quality, well-characterized, standard biological parts. By analogy, one way to think about this would be to look at the transitions that took place around sequencing of DNA. When sequencing was invented, say, in 1977 by Fred Sanger, folks then began to immediately sequence DNA in their own labs, right, using very high cost reagents and highly skilled labor to sequence amazing DNA. Over time, though, it became apparent that it would be better to have professionals, in part, running facilities that would sequence DNA in a much more efficient, much more accurate, much more cost-effective basis. Those became the genome centers. So could we do something like that? Not to replace iGEM and the wonderful diversity of parts. Who knew that you could make little protein balloons? But to complement that network of people and make much more high-quality parts to strengthen what's already happening. So here's the proposed workflow for such a facility. You would have a design team. And this design team is not meant to design every new genetic component from scratch. Rather, as you'll see, it's meant to interface with a community of designers and users, the people who want to make use of the parts, to help take the things that people already know about and make them easier to work with, more standardized, if you will. The BioFab would then coordinate with external suppliers of genetic material, DNA synthesis companies, gene synthesis companies, and so on. We wouldn't want to recapitulate and recreate within this organization the entirety of the DNA and gene synthesis industry. The initial constructs so produced would be subject to a first degree of performance screening. Are they going to work or not? And throw away the things that don't work, since we won't have perfect design tools. As needed, work with external partners to redesign, improve by directed evolution or otherwise the constructs that begin to make it past the first screening. For the things that do so, subject them to a full set of measurements, much like you saw on that data sheet for the cell-cell communication receiver device. Now, while it turned out to be the case that that took about 2 person-years to make that one data sheet, the real meat and potatoes of that work was developing the mathematical data models that would allow for the conversion of, say, the rate of change of a fluorescent protein in a cell to the estimated activity of the flow of RNA polymerase along a piece of DNA. And once those models have been developed for a class of parts, they can be readily redeployed in a more scalable process. Following this would be a documentation team. Following this would be a distribution team that would both promulgate the information via computer databases and a web of registries, as well as supplementing the ongoing distributions a la what's seen around iGEM. The idea would be to build an ecology around this resource, the BioFab, where you could have large communities of users and applications, whether they be in biofuels or medicines or food or what have you. These would be the folks that would be the downstream and upstream customers and designers of the BioFab. In that, these would be the people who would provide the expertise needed to help specify what should be made, and then this organization would execute on the production of those components. Another interesting aspect of this, which is worth supporting, in my opinion, is the idea of having what I'll call an Office of Biological Disenchantment. Oftentimes, when people do a genetic engineering project, it doesn't work. And our current framework, meaning academia and industry, oftentimes does not provide support for capturing and collecting the data on failures. And as a result of not having good data on failures, it's been very difficult to get better at designing standard genetic components that can be put together more reliably. This is something, in my opinion, that has to change. If you think about structural engineering, when the bridge falls down, you go look at the bridge and you try and figure out why it fell down so that the next bridge you build might stay up a little bit longer. And so somehow we might want to consider recognizing and celebrating this work. And if we can't do it via the social engineering of academia, we might have to recognize creating a professional staff that can help us with this. Over time, by doing this sort of work, my expectation would be that we would develop better design rules that would help us to standardize the genetic elements, the boundaries between them, such that we could better support reliable functional composition, meaning when the two elements are put together, will they behave as you expect? I really wouldn't know how to propose doing this today systematically for all the different types of genetic functions we might hope to develop and share. And so this would be a second stage of this operation once it gets off the ground and has some initial experiences. To give you a sense of what this would look like as an organization, you would have user groups, and there would be a set of advisors drawn from the community who would be thinking both about the applications of synthetic biology and the technology needed to make the components. And these folks would together define what the biofab should be producing for the community. Once these projects are identified, they'd be set into the fab via a pilot team. Again, a set of experts drawn from the community that would help with the specifics of the biology and designing the componentry, the screens and selections that would be needed to find the right things that actually work, the measurement technologies, and how to scale up this particular project. That would then couple into some pilot teams, that would involve folks as professionals at the postdoc level and also research technicians. And for each type of team coming in, you could imagine having a project trying to look at control of protein degradation, for example, control of mRNA stability, and so on. Each of these would be independent teams leveraging the resources of the downstream resources, which would include the transition teams, the informaticians, the designers, the measurement teams, the analysis folks, the documentation teams, and the dissemination teams. To put this in context, if you again compare what this might look like relative to a sequencing center, with DNA sequencing, the data type that is being produced by the facility is relatively simple, A, T, C, G, and the quality of that sequence. In this case, we're trying to produce a multitude of genetic functions that have, in often cases, very different activities. Turning on a gene, turning off a gene, having a protein go into one volume inside the cell, or so on. And so this challenge in sort of organizing this is to imagine having a core team that can really scale things up, yet interface with pilot teams and outside users that might be sort of from a biochemical and application case quite specific. In aggregate, I won't go through this in detail. It looks like this is about a 29-person operation, the pilot. In getting this started and doing a lot of groundwork, we now have a landscape where Stanford, Berkeley, LBL, Alberta, UCSF, MIT, and Arizona State, as 7 institutions from North America, are keen to participate in this. Figuring out exactly how to structure this. Is this going to be $250,000 from each university or in-kind donations on an annual basis is something that's incredibly challenging to work out and has not yet been resolved. The obvious thing to note is this would be meant to be an inclusive operation. And so if you'd like to see UST or another institution involved, the Venter Institute, that would be terrific. Another thing that's not yet been well worked out is what the relationship should be between this biofab and industry, right? So if you have some notional company, Engineered Virus Insertion Library Incorporated, EvilCo, how would they make good use of this production facility, especially if the production facility was producing genetic reagents into the public domain? Could you build a next generation of biotechnology that existed on an open technology platform and didn't depend absolutely on exclusive ownership of small numbers of genetic functions? From talking about this with folks like Larry Lasky and others, It seems like there's a way to do this whereby the product of the biofab could be in the public domain, yet the users of the components could protect some aspects of their downstream systems without giving everything away. So that's still to be worked out, and a lot of importance in making sure that the efforts here are compatible with the ongoing investments in biotech. In other words, what we'd like to see happen is an ecology of industry develop around this resource such that they might ultimately support the operations going forward. So here to end real quick, and I did go a little bit longer than I estimated. What's the best funding model for this? Coming back to some of the comments from our last session yesterday. Is this a public-private partnership? Is it public only? Is it a philanthropy? How do we actually get this going? And then what should be the license of the products? Public domain? Or should it be on a contract basis giving exclusive rights to the particular users? Thanks very much for the opportunity to present this project as it's evolving, and happy to discuss this in the little time we've got now or over the rest of the meeting.
Thank you very much. We have time for a couple of questions.
Drew, we've talked right here. We've talked about this before, but how would BioBricks, as it exists today, fit into this model? And why wouldn't you start there?
Yeah.
And improve its parts library and turn it into the biofab as opposed to starting outside of it?
Yeah, I think that's exactly right, Jack. I think that what this thing should be producing is BioBrick parts. The role of the BBF, first and foremost, is to develop the legal framework that would support an open collection of genetic functions that can be used by everybody, right, in a way that's compatible with investment and returns on those investments. And so I think that's what the plan should be. If you noted in the organizations, there's all these research universities, but underlying that is the BBF, right?
Drew Pettigrew from Australia. Great talk. I mean, there are a number of large international consortia on genomes, looking at various cancer genomes, a number of biomarker discoveries, so it seems like you might have the next round coming to put together a major international consortia to work in the area you described.
I'd hope that's true. Perhaps being a little bit early on some of these, it's absolutely surprising to me that for much of biotechnology, there are not what I would recognize as the needed consortia, whether they be industrial or international. Again, as something to single out, thinking about our construction technologies, it is mind-blowing to me that I know of no roadmap, yet alone a funding program supporting getting better at building DNA. This is obviously a technology that's more important than silicon wafers, and it's not that silicon wafers are unimportant, right? The fact that we somehow, over the first 35 years of biotechnology, failed to develop such a roadmap is borderline criminal, given the potential of the technology. And if you dig into it, it's interesting to talk to the team at Applied Biosystems, which made a lot of the early DNA synthesizers building from the 82 Carruthers chemistry. So far as I can tell from my own private conversations, they shut down their DNA synthesis team in 1989 because they figured they had solved the problem of DNA synthesis. That's obviously not true in hindsight. So I'd like to imagine that the world's ripe to make something like this happen, yet it's been surprising to note that there's been a failure in well-organized technology initiatives supporting biotech, at least in some aspects.
I have a question.
Oh, gosh.
I need some of those parts for something I'm building. And so do you supply it simply a sequence?
Yeah.
You don't actually supply a physical piece of DNA?
So the registry of parts that operates out of the Computer Science and Artificial Intelligence Lab at MIT will supply the parts both via computer database, so you can download the sequence, but also as a book with pieces of paper, and on each piece of paper is spotted 100 dried down bits of DNA as plasmids, right? So you can get them both as information free download off the web and via a book. right? That's available at cost, right? Where the cost is the cost of printing. Yeah.
So I need a half a dozen promoters and a few terminators, that sort of thing.
Yeah, and happy to give those to you for your consideration. Something that's important to work out is if you wanted to take these into a commercial application, the downstream freedom to operate.
So is there a lot of red tape?
At the moment, not being the distributor of these components directly, what I can say is I know of no paperwork involved whatsoever. Now, in large part, that depends on one's belief in the research exemption.
Right. Okay.
Just a short question. If, for instance, somebody has a very good promoter, which is multi-copied, singular, something with fancy thing, how you introduce it so it's introduced as another brick, BioBrick? How you do that?
So what you would want to do would be to make the DNA available in a format that was compatible with Tom Knight's standard or one of the more recent standards that's been developed around the BioBricks framework. And these standards basically support how things are connected with one another. So all it would require would be go to the registry, get an account, upload the sequence information, make any small modifications as needed so that it's compatible with the rest of the collection. That would be the easiest way to do it. Another easy way to do it would be to send an email to one of the iGEM teams, either local to where you are, and I believe there is a team from Wisconsin, or somewhere else and say, here's this amazing thing. Why don't you turn it into a part and help everybody by getting credit for doing that?
Okay, thank you very much.